Like the other presenters, he's also done a very good job of seeking out opportunities outside the classroom to learn about marine biology. So 2 summers ago, he did a research project on understanding the three-dimensional structure of coral from Jamaica. Who was the coral? He wasn't in Jamaica, the coral was from Jamaica. And then last year, he did a research project in Western Washington with a dove all summer, collecting information on pinto abalone, and presented that research in many last October. And the current project he's finished is working with marine mammals, dolphins in particular, as you'll hear. And it looks like he's going to be able to present this research in Florida in the fall at a marine mammal conservation conference. Thank you. So when he's not doing marine biology, he's on the track and field team here at Plant Mama, and hopefully we'll go to nationals again in the decathlon. So he has many interests and kind of a dual life here at Plant Mama. So I will let him give you the title of the talk because we just changed it, and I don't remember what we ended up with.
Thank you. Flip the lights real quick. Does this make any difference to anyone, or is it better? Better? Okay. We're going to go with that then. All right, so thank you everyone for coming out to listen to my honors project. So again, my name is Hester, and I'm really interested in conservation, mostly of marine environment and marine animals. And so my project looked at the measurement of trace metals in selected tissues of healthy and unhealthy long-beaked common dolphins, which are also known as Delphinus capensis, in the Southern California Bight. So my committee members were Dr. Van Anderson and Professor Kerry Sevenbergen, who are both here today, and also Dr. Hal Goforth, and he was unable to join us because he's running the Boston Marathon for like the 35th consecutive time or something ridiculous like that. So, first I'm going to start off with giving you a brief outline of what I'm going to be talking about today. So first I'm going to go with the description of Delphinus capensis and talk about why these animals are important. And then talk about trace metals and also why they're important, and then go into the different objectives that I had for my project and the methods that I used to achieve those objectives, and then go over the some selected results and the discussion into future studies. So to start off, there are 2 different species of common dolphins. There's the long-beaked common dolphin and the short-beaked common dolphin. So by the name, assuming you can figure out how they're differentiated. I'll just like to keep things simple for the most part. So I focused on the long-beaked common dolphin, and both common dolphins have this really distinctive white pattern behind their eye. And these animals feed mostly on sardines, anchovies, small bonitos, and squid, as well as what's called the DSL, or deep scattering layer. So that's just a mass of organisms that migrate upward in the water column at night to feed and do various other things. So these animals are able to dive down to around 200 meters and feed on that deep scattering layer. So the size range is about 6 to 8.5 feet, or for people like Natalia or the rest of the world, it's about 2 to 2.5 meters. These animals are coastally located, and so because of that, they're susceptible to what's called domoic acid poisoning. And domoic acid is a nerve toxin that is produced by harmful algal blooms. And so things like red tide, I don't know if you've heard of those before, but they happen because of increased urban runoff, a lot of nutrients in the water that causes this mass explosion of algae, basically. And so those are becoming more and more prevalent over the last few years as there's been more urbanization of coastal environments and climate change as well. And these animals are also found in social groups from 100 to 500 individuals. They can also be found aggregating with other species of dolphin as well. So why study these species? Why are they important? They're the most abundant cetaceans in the Southern California Bight. And so a bight is basically a geographical bend in the coastline. So the Southern California Bight begins right up here on Point Conception and goes all throughout that area, so that's all considered the Southern California Bight. So, like I said, there's been a growing coastal urban population that produces risks to these species. The Marine Mammal Protection Act was enacted in 1972, and that protects any take or any fishing of marine mammals, and so it really protects them. But there's also the commercial and recreational fishery, which employs things like drift nets or trawl nets, gill nets, things that can entangle these dolphins, and then they end up drowning because they're not able to reach the surface. There's also habitat degradation due to pollution, so industrial runoff or urban runoff, things like that, or introducing more amounts of chemicals and trace metals into the environment. There's also the effects of ship operations, like noise and pollution. So an example of this that's going on right now is down in Peru in South America. There's about 3,000 dolphins and porpoises that have been stranded because of ship sonar that was searching for oil. And so it's had a very negative impact on these animals. All right, so how many of you have seen a sign similar to this before? You know, don't eat these fish because there's metals or there's something wrong with them. So I think this is important because it shows that it's having an effect not only on marine mammals but on humans as well. And it's not something that's far away. It's local. This I found in Shelter Island. Took this picture, and so they have detrimental effects on humans too. So the metals in pregnant women, they're often advised not to eat certain fish or any fish at all because they have possible mercury contamination, and mercury is able to easily transfer across the placenta and have devastating effects on the development of the human fetus. And also another example of the detrimental effects it has on humans as well was in the 1950s and 1960s in Minamata and Nagata Bay in Japan. So there was a chlor-alkali plant that was introducing large amounts of mercury into the water, and the fish were found to have about 2,000 times higher concentration of mercury than the surrounding water. And what happened was the people ate the seafood and became severely ill and even died because of it. So I think it's just important to know that it's not just marine mammals that are affected by this, but us as well. So I've talked about trace metals a little bit, going more in depth into what they are. So these are metals like mercury, selenium, lead, iron, copper, zinc. They're metals that are, some of them are essential for normal biological function, things like iron and zinc, but there are also some that are non-essential that can be toxic, and even small concentrations, things like mercury or cadmium or lead. And so these metals, they occur naturally in the ecosystem with variations in concentration. So an example of this is the Mediterranean that has naturally high occurring amounts of mercury, and also the Arctic, which has naturally high amounts of cadmium. And so looking at different marine species, you get different variations, partially because of this naturally occurring amount. But because of this naturally occurring amount, the marine mammals have adapted incredible ways to deal with it. And they have these detoxifying mechanisms that are able to deal with the potentially toxic effects of things like mercury and cadmium, and I'll get into that a little bit more later. But there's also anthropogenic sources. These are sources that are man-made, like pollution, that have been introduced to the ecosystem. And so with these anthropogenic sources, they're starting to have to deal with higher amounts of toxic metals than they've ever had to deal with before, and it's increasing at an alarming rate. So why these are important is because of something called biomagnification, which is the increase in concentration of a substance throughout a food chain. So for example, I'm going to use mercury. So these animals here, they eat a certain amount of food that contains a low amount of mercury. But as you move up the food chain, the next level has to eat more and more because it's a larger animal. It needs more food to sustain its normal functions. And you continue on up the food chain, and each one accumulates more and more, which is denoted by the red amounts in each of them. And that's the other problem, is that things like mercury, they don't degrade, and they don't degrade easily, and they're not excreted. So they just accumulate on up the food chain. A famous example of biomagnification occurred or was talked about in the book called The Silent Spring by Rachel Carson. And this was written back in the early 60s, and it was about the use of the agricultural pesticide DDT and how it was affecting the birds in the area, and that one day the accumulation of DDT was going to cause massive die-offs of these birds, and that we're one day going to have a silent spring, there won't be any birds anymore. And so that book was really one of the things that jump-started the environmental movement. So I decided to focus on a few different metals that I found were mostly focused on within the literature. These metals usually have the highest toxic effects, or in the case of selenium, detoxifying mechanisms. And also these were the ones that were found to have the highest variation as well. And so mercury is found in multiple forms. It's found in an inorganic form as well as methylmercury. And methylmercury is the most toxic form of mercury. And it's been known to be an immunosuppressant. So with high amounts of mercury in the body, you aren't able to fight off infections or diseases as well as you should be able to. The second one I looked at was selenium. And selenium was found to be necessary for detoxification, or what they think is a detoxifying mechanism. And so what it does is it is able to complex with methylmercury and hold it in the inorganic mercury form so that it is no longer as detrimental to the animal as it was previously. And so that complex is called tiaminite, and it's usually found in granular form in the liver. And then the last one I looked at was cadmium. And so this cadmium is mostly concentrated within the kidneys, and it can be detoxified with what are called metallothioneins. And so metallothioneins are able to bind cations like mercury and cadmium and hold them in a state where they're not going to have as much of a toxic effect on the animal. Then you get a little context of the study of what I did and what was already done once I entered into this project. So I worked with NOAA and the Southwest Fisheries Science Center in La Jolla. And they have a specimen tissue collection that goes back a couple decades. And so I took 26 individuals that had, the tissues had already been prepared for me. So the process usually goes, a dolphin is found dead, and they go out, pick it up, take it back to the lab, and do what's called a necropsy, which is the animal form, the animal form of an autopsy. So they find out, how it died and things like that. I'll talk a little bit more about necropsy in a second. But then tissues are taken and stored in a freezer for later analysis if people want to. And so I was one of those people. I wanted to do that analysis. So they have this tissue collection that I looked at, and they had large amounts. So I looked at 26 individuals that 11 were healthy and 13 were considered unhealthy, and then 2 were undetermined. And also the funding to do my part of the project was provided by NOAA to do what is called subsampling, and I'll get into that in a second as well. So for the collection of data, I was not involved in any of the collection of data for strandings or necropsies in my project. It had already been done for me, but I was able to partake in a stranding and a necropsy to see just how the process worked. And so this was one of them. This was a net entangled animal that had died down by the Punoma navel area. This was a young male calf. As you can see, it has a lot of lacerations on the face and missing skin, which was due to trying to get out of the net. And there were also stranded animals that I was able to help out on a stranding. This was a 7-foot, 240-pound male that we found down around the Coronado area that had stranded itself. And I should mention also that this animal was Terciops, so it's not the species I was studying, but it was generally the same idea of the necropsy. This was Delphinus capensis species. So 24 of my specimens were found stranded in San Diego County, and then 2 were found Los Angeles County. Each green dot represents an individual specimen. So let's talk about necropsy a little bit earlier to give you more details on that. So a necropsy is a thorough examination of the body and the organs. So you take measurements like the length, the girth. You go in and get weights of the brain, the liver, kidneys, different organs like that. And you also take pieces of those tissues to send off hormonal analysis and histopathology. So the histopathology looks at the disease within the tissues and if it's present or not. And that was really how we determined whether our species or our specimens were unhealthy or healthy. It was by histopathology. And then also they did MRI and CT scans on some of the dolphins, but they usually didn't do it. It was only if they thought it would produce interesting results. Sometimes dolphins were found with bullets in their skull because fishermen would catch them in their nets inadvertently and then either put them out of their misery or out of frustration would shoot them. So it's kind of sad, but it's not done on all of them. So the objectives for my project were to determine the concentrations of various metals in the liver and kidney tissues of Delphinus capensis that were collected in the Southern California bite. My second objective was to compare the trace metal concentrations between healthy and unhealthy individuals to see if there was any correlation between high concentration of metals and disease, and then also to collect baseline data that would provide important information for the evaluation of Delphinus capensis in the future. So this was one of the first studies done in the Eastern Pacific on Delphinus capensis, and actually I think one of the first done in the world as well. There was only one other that I found done in Korea on Delphinus capensis. So I think it's important to add to that data set. So I had two hypotheses. The first one was that there would be a significant difference in the heavy metal concentration between the diseased and non-diseased groups, thinking that the unhealthy group would have the higher concentration of trace metals. And my second hypothesis was that there would be a significant difference in the toxic trace metal accumulation between mature and immature dolphins, with the mature ones having a higher concentration. So this kind of goes back to that idea of bioaccumulation. The older animals have longer to accumulate these trace metals, and so that's what I based that hypothesis on. So this is the part that I jumped in on, was the subsampling. And so I got the tissues out of the freezer, and approximately 10 grams of each tissue was taken. This here is a liver that I was preparing, and the samples were prepared under sterile conditions and then sent off to a lab at UC Davis for the trace metal analysis. And the lab at UC Davis is a pretty standard lab, I guess, for doing trace metal analysis for any marine mammals along the West Coast. So it was a standard procedure that they use to analyze for trace metals. And here are some of my results. As you can see, the mercury mostly was found in the liver, methylmercury also, and selenium. And so that makes sense with the idea of tiaminite, the selenium complexing with methylmercury to hold it in the inorganic form. And then cadmium was mostly concentrated within the kidneys, is what I found. And selenium and also methylmercury were not detected in the kidneys. All right, so this shows the differences between the healthy and unhealthy individuals, the mercury concentration in the liver. As you can see, there's quite a bit of variation, as noted by the standard deviation bars there. And also the healthy individuals actually had the higher mean. So wasn't what I expected, and I guess that's what happens in science. My sample size was somewhat small, so that could be somewhat of the reason for it. And also in the immature individuals, the healthy mean was higher than the unhealthy, but again, you just have this large amount of variation, so nothing conclusive really there. And then to show evidence for the tiaminite formation in the liver, this graph shows that there was tight correlation between the mercury concentration and the selenium concentration. As you can see, as mercury goes up, selenium goes up as well, and this was found to be statistically significant correlation between the two. So for accumulation by age, the comparison of mercury in immature and mature specimens was done again, and it was found that the mature population had a significantly higher mercury concentration than the immature population, and there was about around age 8 or 9 most of them on the right side of this are considered mature, but maturity and immaturity was determined by gonad weight, not by age. So as you can see, these are overall much higher than this cluster here of the immature individuals. So to revisit my objectives, they were to determine the concentrations of those various metals, and also to compare them to see if there was any correlation between disease and high metal concentrations. And then lastly, to collect that baseline data and to add it to the data set that is mostly non-existent, but there's some out there, and so that will be important for the future of the animal. So my first hypothesis was not supported, the one that I thought there would be higher disease in the higher trace metal concentrations. That could be because of low sample size or just a large variation of things. My second hypothesis, that accumulation would be higher in the older animals, was supported, and again, biomagnification, it just supports that idea. And then also the formation of the tiaminite complex in the liver is showing that they have that detoxifying mechanism, or that selenium is able to hold mercury in a less toxic form. So some possible future studies. Again, I said earlier I talked a little bit about domoic acid, and two of the individuals in my study had very high mercury concentrations, and it was the two that happened to die of domoic acid toxicosis. So it could be interesting to look at what effects that has on the metal uptake of mercury and cadmium and other trace metals like that. Also, it could be a synergistic effect of metals. Maybe it's not just mercury, not just cadmium, but the whole panel is causing health issues in the animal, and so that would also be interesting to look at. And again, the limits to the detoxification mechanism. We don't know if there's a threshold for their ability to detoxify mercury or methyl mercury, and so it would be good to see what the range is that they're able to function in, and also we don't know what kind of effects that these detoxification mechanisms are having on them. We don't know if it's taking more energy or anything like that. So those are some more studies that we could look into. So again, I'd like to acknowledge my committee of Diane Anderson, Hal Goforth, and Carrie Summerberg, who provide a lot of support and direction on this project, and also Noah and the Southwest Fisheries Science Center, the group up there, Susan Shivers and Carrie Daniel, who helped out a lot on the lab prep and everything like that, and also Noah for funding this project. All right, thank you.
Questions?
Yeah. Well, I have no idea what those numbers mean, but I mean, it sounds like basically all the dolphins, there's variation, and the kind of the acute form of disease that you noted in the necropsies didn't make a difference, but it seems like it must mean something that they all had, some kind of level of trace metals. But how significant? I mean, can you give us some idea as to what that means that they have those levels of trace metals that?
Like compared to other marine mammals? Is that what you're saying?
Yeah. I mean, is there a level that's considered a safe level?
Yeah, that's what I was talking about, the future study. It would be good to know kind of what that threshold is. We don't really know that at this point. But looking between different marine mammals, there's some pinniped studies which are sea lions and seals that have been done in Southern California. And so the dolphin, like my study, had pretty similar results as that one in terms of the trace metal concentration. But as compared to different regions around the world, it's hard to look at because, again, like the Mediterranean, there have been a lot of studies there because of their natural mercury that's there. And so those usually have a much higher mercury concentration than things like the Eastern Pacific.
And where would you suspect these trace metals are coming from?
The anthropogenic or natural?
Well, I mean, how do you know the difference?
Can
you tell? Is there a signature that tells you?
I don't think so. I don't know. That'd be interesting to look into. But where they're coming from? Yeah, well, I think in the example of the Minamata and Nagata Bay in Japan, they can just measure the effluent of the chloralkali plant and see what's in that, and then measure what's in the water, I guess. And, yeah. Yes?
Yeah, your conclusion that the healthy animals have a higher mercury level, is it possible that mercury is acting as a medicine against pathogens? I know they used to put mercury in paints to kill fungus and stuff.
Yeah.
It could be having a positive effect on that?
It's possible. I mean, yeah, our results weren't conclusive on healthy or unhealthy. It's thought, though, in previous studies that mercury is an immunosuppressive and not helpful in that, but yeah, as far as my study goes, it's possible.
The sign in San Diego Bay, where do they think that's happening? I mean, is that human contamination?
I'm not really sure. I just found the sign down in Shelter Island, but yeah, I would assume because we have quite a lot of naval interaction and quite a large harbor with a a lot of boats coming in and out, and so.
So it'd be more boats than like industry or something like that?
No, I take it back. It'd be mostly industry over boats, but just have those industries like on the coastline too.
The dolphins you studied, were they of different age? And was it that maybe if they're older, there's more contaminants, or younger, less? Was there anything like that observed?
Yeah, so the older animals did have higher levels of trace metals because of the bioaccumulative effect. I think that, yeah, there we go. So from here, these were all the young animals. Here's age, the x-axis. So up until 6, they had a much lower concentration, except for a couple outliers. But for the most part, as they got older, there was some more accumulation. These two, I believe, were the domoic acid toxicosis, so they had the highest levels. And again, that would be a future study to see what kind of effects that's having.
Any other questions? Okay, thank you.